Hi, good morning. How are you today? I thought I'd share some of the neat artwork on the buildings around where I live. This is just pretty cool stuff. There's a whole bunch of other ones too. I just thought I'd share these three. And it's just a neat way to start the day. Um, I'm intelligently dressed. We're going to actually go pirate style this morning. Let's get that on straight. So I think I left you in the bar last time. Where I was sitting and just minding my own business, sipping on my unsweet iced tea, and Simile walks in, parched as the desert sand, sits down with his buddy Hyperbole, and they start drinking like a swimming pool full of beer. There's something else, I tell you. So let's get on to today's lesson. So this is going to be DNA stuff. We're just going to touch the basics of DNA stuff. This is Unit 1 stuff. And most of the stuff that DNA does is in Unit 6, both replication and transcription, translation, gene regulation. Unit 6 is a biggie, right? And so we're getting to that. But here, just the basics of structure. All of the learning objectives and essential knowledge stuff are on this page, but I've got them scattered out throughout this PowerPoint also. So don't worry about looking at all of them right now. So here we have, this is a key idea that DNA does two quite different things, even though the processes by which DNA accomplishes these two things is very similar, right? One is to transmit genetic information that requires copying, right? So the copies can be transmitted to the next generation, right? Those copies store in the sequence of bases that information, the information how to make proteins, right? And its second job is to be used to make those proteins. So this is about replication. This is about transcription, but the details of both of those are in Unit 6. But like anything else, these functional things are due to the structural aspect of the molecule, and that's what this focuses on most. Okay, so this will be quick because hopefully this is old news. So here we have, in nucleic acids, information is encoded in sequences. The sequences of these nucleotide monomers, specifically the base part of them, the ATCs and Gs. Then they're going to also make a little DNA RNA comparison here, just a little bit more in Unit 6. But here is a very common mistake. This is a nucleotide. This is a base. They are not the same thing, right? Don't call bases nucleotides. Don't call nucleotides bases. It's not the worst sin in the world, but it's a very common mistake. So here they say that they might want you to say what a nucleotide has, right? Sugar, phosphate, nitrogenous base, connected by what kind of bonds are these monomers connected by? Covalent ones, right? And the 5' 3' here, and the structure of them with the nitrogenous bases being like steps of the ladder and the sugar phosphate so-called backbone being the sides of the ladder. All that is in the essential knowledge part, which means if they ask you about the structure of DNA or RNA, all these things will get you points, right? So this guy Chargaff figured out this kind of idea that Watson and Crick then uh, correctly put together was a key to the functionality of this molecule, and that is complementary base pairing. This should be, again, simple, but this is why you pay attention to the syllabus. Notice that they have purines and pyrimidines correctly connected to which are which, whether they're bigger or smaller. These are not things they're going to ask you just memorization wise, but if they ask you about the structure and you throw these things in, that means there'll be a point there. The fact that they're in the syllabus means they might end up on the scoring rubric. So very simply, you remember this? Which ones of these are pyrimidines? Pyrimidines has a Y in it. Thymine has a Y in it. Cytosine has a Y in it. They're pyrimidines. Then you got to remember, right, that uracil and thymine are very similar because they both can bond to adenine, and so uracil, even though it doesn't have a Y in it, it's also a pyrimidine. We're really overkilling here, right? And if you remember the way I remembered it, which one of these is smaller, a single ring? The smaller ones are the ones that are the bigger word, pyrimidine, bigger word, smaller molecule. So here we have anti-parallel aspect of its structure, right? And so you can see most clearly in this diagram, and they then describe that with the 5' to 3'. Remember what that means for the sugar molecules, right? This is the number 1 carbon, this is the number 2, this is the number 3 carbon. Since it's sticking out the most at this end, that's why this is called the 3' end. This 1, 2, 3, 4, 5, this is the number 5 carbon, that's why this is the 5' end. And the anti-parallel idea is that on the other strand of this double-stranded molecule, you have that arrangement upside down, anti-parallel. Okay. Even, look, it even says that these are 3 hydrogen bonds and these are 2 hydrogen bonds. This is another detail they'll never ask you which is which, but if they ask about structure and you say this, it might be there for a point in the scoring rubric. So here we focus that the carrying information function of DNA is all about sequence. Where does it say that? Sequence of these nucleotide monomers, and even more specifically, the sequence of the bases, because the sugar and phosphate sequence is always the same no matter what stretch of DNA or RNA you're looking at. So here's another beautiful picture to leave you with. That's right down the street from my house. It's nice. And listen, I don't want you to be shy about communicating any of this that you don't like the way this is done or that's done, and a suggestion of another way. That's fine with me. You know, don't worry, you won't offend me. Like I told you before, I'm used to being rejected, usually by women. And in fact, just a couple nights ago, that beautiful lady with the glass eye, she called and she said, hey, why don't you come over? Nobody's home. So I went over. Nobody was home. I need some more iced tea. So just one more slide that just to leave you with, and hope you have a wonderful day. Oh my.